Charles, thank you very much, and I'm glad to be there with you virtually. Thank you for allowing me to do this way, and I'm sorry for everybody that I'm not actually there with you. Just one of those things where too many complicating factors, but it's great to be able to participate, and so thank you very much. Charles asked me to speak to you today about some of the work we've done with oncolytic viral therapy for glioma, and I think you all have my disclosures there, so I'm just going to dive right in. I just want to tell you what we're going to talk about. You guys know about malignant glioma, so I don't really need to spend any time on that. We'll give a general overview of oncolytic viruses and then kind of a little bit of the early history of what it was like when we first started working on this, talk about some of our initial clinical experience, then move on to current clinical trials, what future directions are ahead, and some trials as well. So I think this is something that those of you who are clinicians will all be familiar with. You have a patient who has an MGMT methylated GBM, undergoes standard chemoradiation, and does well with that, but then she comes back to see a neuro-oncologist because her scan looks like this over here after her 12th cycle, and she her skin looks like it does down here after having initially had a great resection. And the doctor wants to put me on Avastin, she says to you, but he won't tell me if it's been proven to help me live longer. I've done some reading on the internet, and I know a lot more than he does now. You know, we've all had this patient, right? And I think I want to be put on an immunotherapy clinical trial before it's too late for that. So we have moved on to the world of immunotherapy in many other cancers very successfully. Glioblastoma has lagged a little bit. So let's talk about oncolytic viruses first. So there's been a great deal of interest in using oncolytic viruses for treating various kinds of cancer, but in particular malignant glioma. And on the left here, there's a list of different viruses that have been tested. We've concentrated most of our lab work on HSV, although we've looked also at adenovirus and reovirus. But others have been in this arena as well, too. And Great Britain has done some work on 1716. Nino Kyoko at Mass General has looked at RQ Nestin 34.5. And not listed on here is G Delta 47, which has been studied in Japan. There have been some adenovirus studies, and the biggest champion of that has been Fred Lang at MD Anderson. The Mayo Group has spent a lot of time looking at measles virus, and I know Corey Raffel is close to you all, is interested in that for pediatric tumors. And then we've looked at reovirus, as mentioned. The Duke Group has obviously been most forefront and center with this in terms of the media with their poliovirus. And I think that you can make an argument for one or another of the viruses, but they all have the same basic principles shown here, where the virus can actually go in and replicate and produce new antigens and kill some tumor cells and potentially produce an immunotherapeutic type of result. So I first became interested in this area when I was a resident. I took some time off and was in Bob Martuse's lab at the Mass General then, and he said, "Jim, would you be interested in working this project?" And I thought it sounded really cool. So I said, "Sure, yeah, this is a genetically engineered virus to treat cancer." And they actually had a virus that Don Cohen, a virologist at Harvard Medical School, had created called DLS-PTK. And this was originally constructed with the idea that it would be a good herpes simplex virus vaccine. It was aneurovirulent. Well, we started looking at this in glioma cells, and sure enough, it still killed glioma cells. And in various nude mouse models, we found out that you could actually increase survival and inhibit tumor growth, as shown on some of these graphs. So we went on from there and did some trials, and here they are shown. And you can see that the first trial we did was this trial here done with a company called Neurovir. And this study was done at Georgetown. Martuse had moved to Georgetown by that point, wanted to stay involved. And I was here at UAB by that time. And so we actually became involved with another surgeon, Mike Medlock, pictured here along with Bob, and ran this study. We enrolled 21 patients and found that this virus was safe for using in patients, which was the first big thing to find out because nobody had done anything like this. We were actually injecting the virus directly into the tumors, and not at small doses here, from 1 × 10⁶ plaque-forming units, and plaque-forming units simply just means active viral particles, all the way up to 10⁹. So I remember the first day that we treated. It was very exciting. We were all gathered in the antechamber of the operating room, and this first patient was actually treated at Georgetown, but I flew up there to be there, and there was a great deal of excitement, and I think that there's reason for ongoing excitement today. But we went out and showed that this was safe in this patient population. It doses up to 3 × 10⁹ plaque-forming units, and we had some patients that really had some remarkable responses. We then went on at UAB and did this second study, the middle study pictured here, and this was a phase 1B study. We only had 6 patients in this study, but we got permission for the 6 patients to actually inoculate the virus and then come back 2 to 5 days later and then resect the tumor. I resected the tumor, and then I re-inoculated the virus into the tumor cavity after the resection. So they actually got 2 doses of the virus. And we found out 2 things from this that were important. One, that we could give the viral safely in multiple doses, albeit close together. And 2, that we could actually inject the virus into tumor-infiltrated brain as opposed to just injecting the virus directly into solid tumor. The other nice thing about this was that it allowed me to take some of the resected tumor and do further studies on it. Now, we only injected about 15% of the total dose of the virus into the tumor that we were going to resect, but still we got some idea of some biologic endpoints from that, and I'm going to talk about those a little bit more. Then in the third column, you'll see our third phase 1 study that we did with GTO7, and this was inspired by the person pictured here, Ralph Wexelbaum, who's a really brilliant Ludwig Fellow at the University of Chicago and chair of radiation oncology there. And Ralph discovered that you could actually treat the patients, or preclinically treat other animals that have been treated with the virus, and markedly increase the effect of the virus against the tumor when you gave them a sort of a 1-2 punch. And so we went ahead and did that in patients, and we found out that that was safe as well, and it also was promising. So here's that second study. I'm going to go back this was our very initial study just getting published, and that I mentioned earlier. And then this is the study that we did with our GTO7 plus radiation. This was funded in part by the NIH, in part by industry. And in this, we took 1 step back from our first dose of virus and simply used 1 × 10⁹ plaque-forming units. And then I injected it in 5 sites within the tumor, and then took the patients and gave them radiation with 5 gray the day after. I have the Gamma knife picture here because originally we were going to have a second tier of patients that got treated with 15 gray because we thought that would be a nice dose. While radiosurgery hadn't been found to be effective in recurrent glioma in the RTOG study, there had been some patients that had clearly helped, and we thought, well, maybe if we gave it with the virus, we'd see some effect. But unfortunately, our company ran out of, our biotech company ran out of money, so we just got to do the first dose, but that was fine. We still learned some interesting things, and the virus was safe with 5 gray, and we saw some nice results. So this just shows these conclusions, that we had some impressive radiographic responses with this, and it certainly seemed like something we could study further. Here's some of the patients from that study, and you can see the example. Here's a patient whose disease was growing rather rapidly and had some diminishment after the treatment. And as you know, 5 gray doesn't touch a GBM, so any response here you see is from the synergy between the 2 agents. Here's another patient who had a larger GBM treated, and you can see it almost looks like I went in and resected the patient, but that did not occur. That was all from the combined treatment of virus and 5 gray. Here's a patient with another nice response, and here's a patient with a tumor right in the motor strip that we couldn't operate on effectively because of that. Went in and injected with the virus and got this fabulous CR here. So from the middle study pictured, we did get some tumor study, the tumor tissue that we could actually look at. And here you see some of these samples here. We first looked at immune response to GTO7, and we saw that if you looked at CD3 cells here, which are lymphocytes, you can see that there weren't very many before treatment, but that after treatment you had a marked influx of CD3 cells. And here's an example of looking at CD8 cells, similar kind of finding, not very much pre-treatment, but post-treatment we had these foci of CD8 cells. And this is very important because, first of all, these patients are really immunodepleted. These are patients that were all recurrent tumor patients, and so they, not only have the IL-10 and TGF-beta and so forth from their tumors, but they also have been treated with chemoradiation. When we actually, on these patients, we also did delayed type hypersensitivity testing with 10 different antigens like mumps, candida, and so forth, and not a single one of them was able to mount a DTH type response. So systemically, they're really immunodepleted as well. But nonetheless, you can see that in the brain, we were able to get a lymphocytic response in here with these patients. This looks at some of the other, we looked for lymphocytes in general, CD8 cells. B cells really weren't a player either before or after treatment. And then, not surprisingly, tumor-associated macrophages and microglia also were upregulated. When we went back and looked at individual gene expression and did some KEGG analyses as well as RNA-seq, we saw that most of the genes that were upregulated, particularly in the responders, were immune response genes, further supporting our concept that this was really an immunotherapy as much as anything. And here's an example of some of the biologic processes that we saw upregulated in patients that did better after treatment with GTO7. And you can see that these are immune response, defense response, inflammatory response, responsive virus, et cetera. So this really is, looks like what's going on in the patients that are having the good responses to this treatment. We've gone on since then and done some RNA-seq and studies, and you can also see that in these studies that cytotoxic markers like granzyme B are upregulated, MHC2 is, interestingly, some chemokines, various transcription factors that produce inflammatory responses, and not surprisingly, over here you can see that a lot of inhibitory molecules are upregulated as well, as would be anticipated with any kind of immunotherapy. So we wanted to understand a little bit more about the immune response, and in particular, we wanted to do this so that we could augment our anti-tumor response by further engineering the virus. And so we made viruses that actually expressed a variety of foreign proteins, and we also looked to see if we could increase the replication of the virus, because I have to say the first virus that we used, GTO7, had 2 sets of deletions. So it was actually a really wimpy virus. We chose it because neither Bob Martuzza nor I wanted to be known as the crazy guys who caused a bunch of encephalitis and deaths in people, and also, more importantly than that, we didn't want to set the whole oncolytic viral therapy field back years by doing this. So we were conservative, and we still had some interesting responses, but we felt at this point that we could afford to be a little bit less conservative, and so we wanted to see what we could do to increase the replication of the virus. And finally, what other concurrent meds could we give with the virus to, I hope, increase its efficacy? So I'll talk about that in a little bit, but I do want to finish off talking about GTO7 before we go any further. And so GTO7 has gone on and been studied by Greg Friedman, who's pictured here, an oncologist, pediatric oncologist here at UAB, and Jim Johnston, who is a pediatric neurosurgeon here at UAB. And together we developed a protocol to treat patients with this who have recurrent tumors. This was based upon some work that Greg did that showed that the pediatric glioma xenolines were actually even more sensitive to the virus than the adult gliomas were. And he also looked at a variety of other pediatric tumors that are important, such as PNETs, which we don't refer to anymore, but ependymomas and so forth. And what he found was that the tumors were much more sensitive to killing by the oncolytic herpes virus than the adult GBM specimens when looked at in vitro. And he also found that glioma stem cells from the pediatric brain tumors were sensitive to killing by these viruses. So we went on and together developed a phase 1 study for children that were 3 years and older to give the virus initially alone and then with an adjuvant dose of radiation. And these were patients that have failed surgery and radiation and have recurrent progressive supratentorial brain tumors. So initially we elected to look at supratentorial tumors because we didn't have any data in the posterior fossa, and we didn't want to cloud the fact that perhaps toxicity that developed there might mean that it was not appropriate treatment for children at all. So in the initial study, we were just looking at supratentorial tumors. And so this included the tumors listed below because obviously kids don't get tumors, brain tumors as much as adults do. And so we had a wider variety of tumors that we were testing, or that at least would be eligible for examination. And of course, this is true in most pediatric oncology studies. So nicely, we found that we're now reached this third level of enrollment here in the 1 times 10 to the 7th plus 5 gray of radiation arm. The virus is being inoculated in up to 4 loci. And we've had some really nice responses. Here's an example of an early response, and you can see just immediately after inoculation, you're getting that virus, oncolytic virus type of replication that's producing direct necrosis or hemorrhagic necrosis within the tumor. Here's another patient that was treated, and you can see that this patient has done really well. He had a KPS that was 80 prior to treatment, and then after treatment, it improved to 100 by 12 months. And now you can see he's had this nice response as you look sequentially across here. He hasn't had a CR, but he's had a marked decrease in the amount of enhancement and edema. And we do know, just as an aside, that a lot of our patients seem to have this sort of Swiss cheese finding on their MRI scan, which you can see these little holes in the virus and the tumor developing. And the import of that yet is not known, but we certainly have seen that in multiple occasions. If you try to figure out, as best you can from a phase 1 study, whether this is having an impact or not, if you look over here in the left column, you can see that if you collect a bunch of studies of pediatric malignant tumors that are recurrent, median survival looks like about 5.6 months if you look at all those. And right now, granted in our small study, we have a median survival of just over a year in similar patients. So I think for what it's worth department, that at least looks like we need to continue to study this approach in children. So now to get back to my thoughts about increasing the efficacy of GTO7 in adults by trying to improve the immune response against the virus and tumor, we developed a virus in our laboratory called M032. And M032 makes interleukin 12. IL-12 is a critical cytokine for producing both T cell and NK cell responses. The T cell responses can be memory responses, and they can, we hope, reinvigorate an immunosuppressive tumor environment. And up here you can see an example of one murine model where we treated with saline alone, and then we treated with the murine equivalent of M032, which is called M002. And you can see there's a marked improvement in survival with this. And when you look at, take these tumors out of the mice, so these are all orthotopic syngeneic models, and you look at the immune cell infiltrates, you see, interestingly, that even the parent virus produces some increase in immune cell infiltrate. And if you look at these 3 graphs here, I'm trying to get my pointer here, the blue represents mock treated, the purple represents treatment with the adult virus that does not, or the parent virus that does not express IL-12, and the yellow expresses is a virus that expresses IL-12. And what you can see is that after about a week, there's a marked increase in CD4 cells and CD8 cells in the tumor, as well as NK cells, with both viruses, but particularly with the IL-12 virus. But then this response sort of dies off over time. When we looked at other preclinical models, we saw that our virus, the IL-12 virus, was able to replicate better, and that's due to some changes in the genetic engineering of the virus. But if you look, this is the IL-12 virus in vitro looking at replication, and this is the parent virus, and you can see there's a marked improvement in replication. when we compared the IL-12 virus to another virus, like an IL-4 virus or the parent virus in our various preclinical models, we saw that the IL-12 was the best performer. And then we went to AOTIS, which is a hypersensitive primate model for looking at herpes simplex encephalitis. We saw that after we gave the IL-12 virus, there was in fact some inflammatory response, which is pictured here, which is exactly what we wanted. But what we didn't see was neuronal dropout when we looked at these AOTIS brains later on. So I think that was exactly what we were hoping for, an inflammatory response that could be used against the tumor, but no neuronal loss on a permanent basis. So we developed this clinical trial for treating with the IL-12 virus, and this is actually underway right now. And we've treated 7 patients to date and have gotten as high as 10 times 8 to the plaque-forming units. So we hope to get to a proposed recommended phase 2 dose level and treat 10 more patients at that level. Pictured here are some of the people who have been very instrumental in this trial as well: Dr. Gillespie, Dr. Chambers, Dr. Riley, and Jinmei Leavenworth and Larry Lamb as well. Just to give you the sort of for what it's worth, here's one of our initial patients treated at a very low dose. They initially had progression after failing Avastin, and so they were enrolled in the M032 trial. And then there was this sort of likely pseudo-progression response pictured here in A that caused an increase in enhancement. So we restarted Avastin and treated the patient with that for 9 months. And then got an MRI scan after 11 months. So the patient was off Avastin for 2 months now. And of course, if you fail Avastin and then restart it, you don't ever get a response the second time. So this clearly had to do with this being a pseudo-progression response. And the patient survived for another 6.5 months even after this without additional treatment. So we found that heartening for our very first patient. And this graph down here just shows that when you looked at systemically for a response, so this is, we don't have permission from the FDA yet to go back and do that nice phase 1B type study where we inject and then resect tumors. So we are dependent upon looking systemically for responses. But even systemically in this patient, we're able to see this nice response of an increase in T effector cells and T central memory cells, as you can see pictured here. So this has been promising as well. Here's another patient from the trial showing their response, and this is a response that we think shows a different kind of response that we see in some patients, where you see this big hole that's eaten in the tumor by the virus by day 28. And we think that this is a patient who's having most of the response to the viral replication of the tumor. It'll be very exciting when we can actually go back in with a subsequent patient group and actually look and see how where we can do resect the tumors and see what we're seeing on an immunohistochemical basis, as well as looking at next-gen sequencing. But if we look at these patients overall, we've had 2 patients that have died really early. Both were being treated at their 4th recurrence, and so perhaps were not good candidates for the trial. But everybody seems to have done relatively well with the treatment, and we have some patients here that are still alive, and we're waiting to enroll our next patients. So we think that our median survivals, again, are going to be close to a year right now, and again, great in this recurrent patient population. We have one of the neurosurgeons on our faculty here, Dr. Rene Chambers, who's pictured here, was a former veterinarian. And you can see her here with her love of animals as well. And she actually became very interested in this because although she's a human neurosurgeon now, she still has an interest in veterinary medicine. And so she has actually developed a study that's been funded through a U mechanism to study this in a consortium of dogs with glioma. And we've actually enrolled patients here. You can see a couple of dog gliomas here. And so far, this does not seem to be producing any toxicity in the dogs as well. Kevin Cassie is pictured here, and Kevin is a collaborator of mine that's worked on this C134 virus, which is a novel virus that actually has part of the CMV genome just completely coincidentally with Charles's earlier described work here. And it's since they're both from the same herpes virus family, both HSV and CMV, one might expect that the genomes would have some similarities. And so the gene that was deleted from GTO7, gamma 134.5, has a homologue in C134 IRS1. And it turns out that this actually allows the virus to replicate better, but does not recapitulate the neurovirulence. And I'm going to move quickly through these more scientifically based studies, but basically these show that the virus is able to produce an acute inflammatory and robust interferon response. And here, this just shows some of the mechanism of how ordinarily 34.5 allows viral replication to take place in the wild-type virus, and how if you replace 34.5 with this IRS1 gene, you still get these proteins made, which is not seen in viruses such as GTO7. Viral recovery is increased in the C134 model over these first-generation viruses, indicating that replication is better, and you can see improved survival in some of these models as well. So it turns out, why doesn't it replicate everywhere? Well, it's dependent on interferon. It still is interferon sensitive. And so one nice thing about gliomas is that most gliomas have interferon alpha and interferon beta gene deletions. So as a result, the virus is still able to replicate here. And here you can see that the wild type virus is dependent upon, it does not care, so to speak, as to whether the cell is able to phosphorylate stat or not by means of an interferon response. But here you can see that C134 still is inhibited by the production of interferon gamma and phosphorylated stat1. I'm going to skip through these just in the interest of time. So we now have developed a study, and the FDA has approved our IND for this particular protocol. So we're just getting this study up and running really probably this quarter of 2019. So we think that oncolytic virotherapy is here to stay. Here you can see that there's actually been one herpes virus that's been approved by the FDA, Biologic, or TVEC as it's more commonly known. This is for use in metastatic melanoma. And as I was talking to Charles about before we started this, there's been some very exciting studies published recently about this. So glioblastoma, as we began the talk by saying it's a potential target for immunotherapy, has not had great results so far in the studies with checkpoint inhibitors that have been done. And this may have to do with the fact that if you look at this figure, which shows the relative burden of neoantigens and mutations across tumor types, with the hotter tumors being over here, gliomas are relatively on the left side of this. They're not as hot tumors as these other tumors are, and less likely to respond to immunotherapy. So what can we do about that? Well, we want to have as many neoantigens as possible. And so one way to handle this is to try to use oncolytic viral therapy to produce an intratumoral T-cell infiltration and produce these neoantigens, as well as produce an immune response against the tumor that then can be carried forward after the viral therapy is played out. And this was this really nice Cell paper that was actually a clinical trial that was published and showed that they had this remarkable response rate of 62% to the combination treatment, with a CR rate of 33% for the combination in widely metastatic melanoma, which I think is very impressive. So we think that you can, in fact, convert a cold tumor into a hot tumor by combining immunotherapies. Using oncolytic viral therapy in conjunction with checkpoint inhibitors seems like a logical way to go. So we're doing a lot of this work right now in our patient population, and I think that, in our laboratory. And we found that, in fact, these checkpoint inhibitors do seem to produce improved responses and long-term survivors in our models. They increase the TILs, and so we think that we're going to be able to take this, we hope, into people, and we're working right now on a clinical trial. We've got a proposal in front of Merck right now to look at this with Pembro, so we're keeping our fingers crossed that we'll be able to use that. We need to continue to use RNA-seq and other sequencing techniques to help us understand the nature of the immune response. I'm doing some work with that with Kevin Cassidy at Nationwide right now, and some of our old specimens, as well as with Genmay 11 work here at UAB. Certainly, we need to see that some of these viruses, like the IL-12 virus and that the C134 viruses, aren't going to have issues with toxicity as we study them in more depth. You see that Martuse's group here showed that they had a version of the IL-12 virus that looks like when you give it with dual checkpoint inhibitors, you actually can have even better anti-tumor efficacy. So that's exciting and will be something we'll be trying to see in our preclinical studies. And Kevin Cassidy has shown that Rux actually can increase the anti-tumor response as well. So we need to try all these combination therapies in our patients, but first of all, we actually have to show that the viruses alone are safe, and we're in process of doing that now as described. I think finally we have to really move this to upfront treatment instead of just doing it in recurrent patients, and I'm hopeful that we can convince the FDA that now is the time to do that. You know, here's an example of the tumor cell load. If you actually do a resection, right, and then you add radiation therapy and then a chemotherapy, you can get your overall tumor cell burden down here, and then we're sort of hitting the patients when they're back up here at this relative secondary peak here after they've recurred. So we want to, or maybe even here, and in some cases after they've recurred additional times. So we really need to try to get them when they're right in this stage for us to have the best chance, and certainly before their immune systems have been burnt out by all the treatment. So this is what's ahead. We got to get this into upfront patients. We need to work on minimizing immunosuppression by chemoradiation and steroids. We need to see, do these foreign gene expression, do they matter? Do they increase efficacy, or is the viral infection alone good enough? And then I think also, are these less debilitated oncolytic herpes simplex viruses? MU32 is less debilitated than GTO7, but C134 and Nino Kyoko's virus, RQ Nestin 34.5, are even less depleted, I should say, less debilitated than MU32. And then again, finally, what's going to happen when we use these in conjunction with a variety of other immunotherapy agents? So we're excited. We think that the future is bright for immunotherapy in general for glioma patients with all the knowledge that we've developed with others. And we think that oncolytic viruses certainly have promise and may possibly be an important player and contributor in the future. This level of work cannot be done without a lot of help, and I'd like to at least acknowledge some of the people that are involved in the clinical studies that you've seen here. And thank you again for your kind attention, and I'd be happy to answer any questions.
Jim, that was awesome. Thank you so much. I have a question myself, and when I gave my talk right before yours, I had mentioned that I kind of got interested in the virus-tumor relationship because of a comment that I heard like a long time ago where someone suggested that when tumor cells become tumor cells, they lose the intrinsic immune function and some of the antiviral intrinsic immune function, which allows them to become vulnerable to a virus, which I guess is sort of the fundamental concept behind this type of therapy. Can you mention sort of your concept of that, and is it due to lack of interferon response, or what is that due to?
I think that's a great question, and I think it's there's different answers to that question. For example, in oncolytic herpes virus, the lack of interferon response is important, and I think that's important for other viruses as well. Adenovirus plays on that whole PRB, the retinoblastoma pathway, and the absence of that in its normal state. This gamma 134.5 deletion that's important for the safety of oncolytic herpes simplex viruses. Actually, is probably the major factor in why the virus is debilitated, but it still allows it to replicate in tumors, because the gamma 34.5 gene, ordinarily its gene product counters the phosphorylation of PKR or its gene product, its direct protein phosphorylation product, EIF2 alpha. And so if EIF2 alpha is phosphorylated by PKR, then in fact, normal host cell protein synthesis is shut off. And so basically the cell suicides so that the viral replication can't spread. And if no proteins are made, viral proteins are not made either. So gamma 34.5 usually interrupts that process and allows the viral proteins to continue to be made in infected cells. If you take that protein out, then in fact, in a normal cell, the viral infection will be shut down. And in tumor cell, because of its lack of an attack pathway, there will be allowed the replication can take place. So it varies a little bit from virus to virus, but certainly that PKR pathway is an important one for many of the viruses.
That's interesting. Thank you. Are there some other questions since he can't see you? Anybody have questions for Dr. Markert? Can you speak into the microphone over there?
Yeah, I have a question. So apart from what you said about the phosphorylation and that kind of tumor specificity, is there another thing that you modify on the virus to ensure cancer specificity, or do you use specific promoters or something, or do you actually care if the virus also infects other cells? So that'd be the one question.
Yeah.
The other question would be, you mentioned suicide programs. Have you considered using suicide genes that would actively, on top of the viral replication, kill the cell, like the thymidine kinase or the cytosine deaminase, or one of these approaches?
So, there were a couple questions in there, and the first question is, we don't target the virus to cancer cells by choosing a specific receptor expression. Now, there are groups that have done that. Gabriella Campanelli's group in Italy has actually looked at that. Reutzmann's group looked at that after I left. And so their concept was, well, can we use a virus that's really essentially wild type and target it directly to like an EGFR receptor or some such thing? And so those two groups have looked at that. To my knowledge, that has not entered the clinic anywhere, but it certainly is something that could be considered. And then your second question about using suicide genes is a good one. I think the HSV that we use actually expresses thymidine kinase. And so we did some experiments with thymidine kinase in ganciclovir and didn't see any additional kill. And I think that's probably because the virus is killing the cell that it's in anyway. And so it doesn't really add anything to kill the cell twice, so to speak, the cancer cell that it's in. So, and then you do, but I think we have also made another virus that, uses that same sort of approach that the Tokogen model does for changing 5FC to 5FU with cytosine deaminase. And again, had some promise with that, but it wasn't as promising as the IL-12 virus, and hence that's the virus that we chose to move forward with.
Well, Jim, I can't tell you how much I appreciate you beaming in from outer space to give us a Deus Ex Machina speech. And I really appreciate it, and thank you again. Good luck with your case later.
Thank you for the invitation, and it's been a real pleasure and an honor, and thank you for including me.